My name is Spencer Sanchez, and today I'll be sharing my project that is focused on fungi associated with sooty bark disease in big leaf maples. I'm currently a student at Tacoma Community College, hoping to gain my Associates of Science in Biology, and then hoping to transfer to the University of Washington for a bachelor's in marine biology. And my community is here in the city of Puyallup. All right, so just to provide a bit of background, the focus of this project is on sooty bark disease, but more specifically in big leaf maples. Sooty bark disease does tend to show up in other trees aside from maples, but the reason why big leaf maples are the focus of this project is because they're the most prevalent and like more common urban tree that we see here in Washington, or at least in the cities. So they do make up a large portion of our urban trees or urban forests and therefore should be given a good amount of care and attention, especially when it comes to a disease that has been on the rise, so to speak. But basically, the idea was to isolate the different fungal bodies associated with sooty bark disease and sequence those isolates in order to confirm that the fungi were successfully replicated in a lab setting, and hopefully use that data to be able to delve deeper into what is causing this disease, or even to be able to monitor the systems as the disease progresses, while also being able to make a link back to urban forestry and focus on the importance of urban trees as well as the benefits of them to our day-to-day lives. So what is sooty bark disease? Sooty bark disease is caused by Cryptostroma corticale or Biscognoxia spp. Depending on the appearance of the fungal growth on the tree you're looking at, that can give you a bit more insight as to which of the two fungi it may be. And what these do is they essentially cause this infection, which tends to appear on the tree stem or its branches. And when you do observe it on the tree, it looks as though the bark has been pushed out of the way and has been replaced with these black, sort of ashy-like fungal spores that tend to fly and be released easily into the wind. Um, and this is seen a lot more commonly, or it is more prevalent in warmer climates because warmer weather is ideal for fungal growing conditions. And the tree as well is usually under more stress due to the fact that it has to go through a longer drought period than anticipated, making them a bit more susceptible to contracting this disease. And so in these pictures to the left, you can see the two different fungal bodies, the top one being Biscognoxia spp, and then the bottom one being Cryptostroma corticale, and just the variety in terms of sooty bark disease. And so the approach and general process of this project and the path for collecting samples was to go out to different parks, those parks being Wildwood, Clarks Creek, and Point Defiance Park, and collect different bark samples from big leaf maples, which were suspected of having sooty bark disease. Then bringing those samples into the lab for plating, and from those plates, make secondary isolates based on the growth. And then from those secondary isolates, we would sequence in order to get confirmation for the identity of the fungus. The materials that were used for sample collection included plastic sample bags, which were used to store the bark samples, a knife for retrieving the sample from the tree to make it easier just to remove a good piece of bark, tape and Sharpie for labeling the bags for each different tree from each different park, along with a date for ease of adding to iNaturalist, then a lighter and alcohol for sterilization of the knife so that you wouldn't cross-contaminate between collecting each sample. And as far as lab materials go, those supplies were provided by the WSU Extension Center lab. And so those materials included things such as glass petri dishes, ethanol, bleach, timer for keeping track of how long the bark samples have to be in each solution, media for plating the bark samples, which in this case was PDA plus S/C, which stands for potato dextrose agar, with streptomycin and chlorbenicol, I believe. That's how it's pronounced. I'm so sorry. And then scalpel tweezers and any other materials that were needed along the way, such as paper towels. And here in the images to the right, you can see the general setup for how the sterilization of the bark samples went. And then from there, the bark samples were plated and left to grow until the decent growth was seen and we were able to make secondary isolates. And so this is a tree equity map of the area that was monitored. The area and parks that were picked for this project was specifically to find parks that were more naturally maintained than an urban or a neighborhood park. So if you've ever been to Point Defiance, that is a great example of a naturally maintained park with tree growth that is less disrupted. And hopefully that would help us get a better idea of how the sooty bark disease manifests and grows in these kinds of areas. And included here is also a map of all the trees that were sampled, save for two of them, due to those being more so samples of interest than trees themselves. So for example, one sample that was collected at Point Defiance was from a tree stump and not a living tree. So that made it a bit more difficult to add to iNaturalist. But it was kept track of, and we know the location in case we would like to go back and collect more samples. And then in this slide, we have the individual score for each park that was visited, as well as the equity score for the nearby Puyallup Indian tribe. just to shine some light on how it is significantly lower than the other areas that we see here. And this can tell us a lot about resources that other nearby communities may not have or may not be receiving, as well as how those areas that do have a higher score and a better canopy coverage can provide shelter in a safe area, which can then hopefully be a push to get those resources out to other communities that don't have it, just by showing the importance of having this canopy coverage, not only for us, but for the ecosystem. And then here's the data that was collected as plates were made, and they were visually categorized depending on what was seen growing. So in the bar graph to the left, we can see each sample. So there were 12 samples in total that we were able to collect from the trees that were found. And from those 12 samples, we see how many visually were showing to be Cryptostroma corticale and how many were something else, which could be something such as Penicillium, Epicoccum, Phomopsis, Aspergillus, or any other unknown fungi or yeast that just didn't look like Cryptostroma. And then here to the right, we can see the Excel spreadsheet. I do have more, but just like for the sake of time and also fitting it into the slides, I just included the one with the isolate number, the plate number, the suspected result, as well as a category to see if the plate was contaminated and whether or not we would discard it. And then comments which were saved for isolates that were just replated to remove any contamination that may have been present. And then this is the fun slide. This is the slide that has all the plates. We can see like why and how they were categorized the way they were. And we did that based off the way they visually looked. And so here, for example, in the top left, we can see that these were the plates that were categorized as having Cryptostroma. We can see they have this pinkish or even can sometimes have like a greener color, and they have this fuzzy white soft layer if you turn the plate around. And the ones that were a bit more on the pink side were suspected to be potentially Biscognoxia spp, but we can't be sure of that until we do sequencing. Whereas, for example, the plates to the top right were Penicillium or plates with other contaminants present that may have also been seen a bit frequently. And then if you look to the picture below these two, you see the plates side by side with the original bark samples, as well as the secondary and any tertiary isolates that were made. And you can see the variety of colors that were displayed. And then something that just to add on here a little bit is this picture to the right. This is Aspergillus, and this was one that wasn't seen frequently, but if you ever find yourself in a lab setting or you see something like this, just keep in mind it's not good to breathe in or be around. So we did not open these plates again. And so moving on to why this is more important and just to delve a bit deeper into the importance of this, urban forests can provide a safe oasis for many people, even people who may not be a part of the community. There can be a lot of disparity in terms of how urban forests are distributed throughout the city, and it may not always be equally distributed due to the fact that different parts of the city may have less access to assistance for, say, management of these forests, and knowing how to care for these trees, along with the lower canopy, just making it a bit more difficult for these areas to provide shelter that they may be looking for. And so bringing these issues to light, we can hopefully provide these resources and advocacy and community engagement to increase the urban forests in the cities. Another factor is the fact that the diseases that affect trees can also affect us. So Sooty Bark Disease has been found to cause respiratory issues to individuals, more so those who frequently handle wood and work in sawmills and handle those tree logs a lot more on a day-to-day basis. And while walking past the tree that has Sooty Bark Disease can't cause these respiratory issues immediately, prolonged exposure could. So potentially, if you could say if this issue continues or even worsens, this may become more of a worry for us. And hopefully before that point, we can get more involved and kind of allocate more resources to this issue. And lastly, strong forests promote biodiversity, and biodiversity can lead to a more sustainable ecosystem. The healthier the urban trees and forests, the healthier we are, in order to be able to carry out these positive environmental effects that trees have for us in terms of providing shade, providing shelter, not only for us, but for animals, being a food resource for them, lowering CO2 and general heat in that area. Those forests do have to be healthy. And just by getting involved and looking at more of these diseases and furthering that data available on known fungal bodies that are negatively impacting the health of vital, important trees, could potentially assist in finding a solution to help these trees recover or even avoid becoming infected in the first place. And so the next steps for this project will be sequencing the secondary isolates that we got just to confirm the identity. Unfortunately, we weren't able to sequence before the presentations, but the good thing is we do have a good amount of data to show from these plates, and we will be able to move forward with sequencing. And the sequencing will just help to show us the genome of the fungus and allow us to really like 100% identify what is growing on these plates. Another thing is visiting more parks to find more samples of Sooty Bark Disease and hopefully add to the current data, and as well as continue working with Washington State's Puyallup Extension Center, working with Mary Ann, who's awesome. She works down at the Puyallup Washington University Extension Center, and she's invited me to help with the inoculation of these trees and just discovering more about how this disease affects the trees in their beginning stages. And the acknowledgements and appreciations. So just to acknowledge and appreciate the amazing funders that made this internship possible, as well as my supervisors, Fig, Joey, and Zay, and Mary Ann for helping me out in the lab and with IDing plates, and as well as anybody else who joined us along this journey to teach or just show us new experiences. For example, like taking us out to parks, having presenters, anybody who was a part of that, thank you so much. This was an amazing experience, and I'm glad I was able to be here and even present my project to you all. So yeah, thank you.